And so I'm trying to figure out if the color affects reproduction at all, because presumably whatever is giving it the color is passed on to the next generation. And so does that affect reproduction?
My project mainly comes in and is looking at how disease is affecting these corals and how that relates to genotypic variation. But that's just one piece of the puzzle. There's this whole slew of other things that are affecting corals. So if this is something that you're interested in, there's definitely many things to be studied and figured out about these corals still.
Everyone, welcome to AP Biology class. Today we've got some awesome friends from FSU Biology again here to talk to us today about coral. So I'll do a quick bit of introduction for both. The style will be that we'll go about 20 minutes each. You can ask questions during or after. Just let me know. First up, we've got Allie Blanchette. She is a fourth-year PhD candidate in biological sciences at FSU. She works in the lab of Dr. Andrew Rossweiler and Dr. Sarah Lester. Her lab studies coral reef ecology and conservation. She's specifically interested in understanding of things that influence coral stress and survival, such as genetic differences and impacts of farmer damselfish. Before FSU, she got her bachelor's degree in environmental science at University of California, Los Angeles, UCLA, and worked for 3 years in a variety of internships and research assistant positions in ecology to figure out what she was most interested in. And second, today we'll have Laura Anthony. She is also a fourth-year PhD candidate. She's at the FSU Coastal Marine Lab studying deep sea ecology and corals. She's advised by Dr. Sandra Burke. Her dissertation research is focused on how new environmental variables and climate change impact the reproduction of reef-forming deep sea corals. She received her undergraduate degree in marine biology at Western Washington University in Bellingham, Washington, which I mentioned one of the coolest places in the world. There she researched chemosynthetic methane seeps. LA is pretty cool too. All right, let's give them a warm welcome.
All right, so hi, I'm Allie. I'm going to be telling you about one of my PhD projects today, looking at coral restoration in Bonaire. So to tell you a bit about myself first, I'm from a town pretty close to Baltimore, Maryland. So to be an obnoxious Marylander, I have to show you our amazing flag. And then I did my undergrad at UCLA, where I did a class research project where we got to study coral reef ecology, and that's what got me really excited about realizing marine biology could be a career and not just like a pipe dream. And then I did a bunch of internships before starting grad school. So this is an example. I was working to study seagrass habitat and oyster aquaculture with the USDA. My favorite fish is a farmer damselfish. I'm not going to be talking about them much today, but one of my other projects is on them. That's that fish over there on the right. All of the bumpy orange branches are live coral, and all of the fluffy fuzzy stuff on the bottom of it is algae, and those damselfish will actually farm little gardens of that algae. They have a lot of personality. They're really cool. So I'd be happy to talk more about those at the end if there's time. I study shallow tropical reef building hard corals, like the ones shown in this photo here. So coral reefs are a really important ecosystem. They provide habitat for a lot of different species. So they have high biodiversity. And they also provide important ecosystem services to people. We get seafood from them. They will also dampen the waves of storms. So as a hurricane's coming through, if there is a coral reef there, the storm surge won't be as bad, and it can protect those coastal towns. But as you probably heard, coral reefs are not doing too hot, in part because the water is getting too hot. So corals are facing a lot of stressors, one of which is ocean warming, which leads to coral bleaching, as well as a whole slew of others, including storms, pollution. This can be chemical pollution, plastics, nutrients. Disease outbreaks are also a big problem. And then the corals can get overgrown by a bunch of different organisms like algae, cyanobacteria, and something called ciliates. And so one of the things that people are doing to really combat these coral declines is coral restoration. And so coral restoration is usually based off the fact that corals can reproduce both sexually and asexually. So you can take a coral like this branching one here, snap the branch in half, and then you have two clones that can continue to survive and grow. So they'll do that a bunch of times, fill this whole nursery tree with those branches, let them grow big and strong until they're big and strong enough to be put out onto the reef. And so when successful, restoration can do a lot of great things. It can increase the coral cover on the reef, so just how much coral there is. It can also increase genetic diversity if the restoration group is intentionally outplanting many different genotypes. Another strategy that they could take is intentionally outplanting genotypes that are locally adapted or have high fitness. So fitness is the ability for an individual to survive and reproduce. Certain genotypes are going to be more fit in certain environments, so they can put those genotypes out that will then have a higher chance of surviving. I give the caveat of when successful because coral restoration is really hard, and a lot of the things that cause corals to decline in the first place are still out there. Like the ocean is still getting hotter. There's still a lot of pollution. And so a lot of restoration groups, what happens is they put the corals out, and then a year or so later they die and they don't really check on them or upkeep them. So it's a bit of combination of like unfortunate environmental factors and then also like different management techniques. And so one restoration group that has had a lot of success is Reef Renewal Bonaire. And so Bonaire is an island in the southern Caribbean where that red pin is, and they have relatively healthy coral reefs compared to the rest of the Caribbean. They're kind of like one of the shining coral reefs that people know about for the Caribbean. And this is due in part to just lucky location. Because they're all the way down here, a lot of hurricanes miss them, so the hurricanes go up here. So they just avoid that natural disturbance. But they also have a long history of conservation. So Bonaire has put some sort of marine protections in place since the 1960s. And currently the entire island and all like the coral reefs around it are listed as a marine protected area. And this is basically a conservation management strategy, which can vary from place to place, but what this means for Bonaire is no one is allowed to do any spearfishing around the island. Hook and line fishing and trap fishing is like a bit more unclear. They say they don't let people do it, but then you see people do it, so it's also like a enforcement issue. And then they're really strict on like you cannot take anything from their reef. You can take pictures, you can dive. It's a really big diving tourism place, but you're not allowed to touch their corals. They're very protective of that. And because of that, they have really healthy coral reefs. And they also have Reef Renewal doing a lot of great restoration work. So Reef Renewal has been operating for over 10 years now in Bonaire. They have 22 outplanting sites. So that's what some of these pins on this map are showing. And they have 5 coral species that they are working on restoring. So you can see 4 of them here. The 5th one looks pretty similar to this one. And so they have a really high success rate. If you were to look at the ecological footprint, which is basically if you were to take all the coral around Bonaire that is there because of Reef Renewal's restoration efforts and combine it into like one compacted area, it'd be the size of 2 soccer fields. And Bonaire's not a very big island, so 2 soccer fields worth of coral is like pretty substantial and exciting. And then lastly, I was talking about genetic diversity and having multiple genotypes. Reef Renewal is aware that this is important, and so they have 8 to 30 genotypes per each of the species that they are outplanting. And then their main primary coral species that they are working on outplanting the most with and have the most restoration sites for is staghorn coral. Its scientific name is Acropora cervicornis. So this is a massive, yes?
I just had a question back to the success rate you mentioned in terms of the area with the soccer fields or the different species or genotypes. Is time or longevity also factored into that? Like, do they actually stay or stick versus maybe some of the other places that aren't as good? Maybe they plant them or anchor them and then they aren't there a year later. Is there like, hey, they're still here for this amount of time, or does that factor in?
Yeah, so the way they calculate this is Reef Renewal is really good about monitoring their restoration sites. They go back every year, even for like the 10-year-old ones, and check how much of it is still there. And so I'll talk a little bit later about this like photography method that they use, but basically they take a bunch of photos to map out where the coral is. So they do that at all the restoration sites. So this stat comes from their 2022 annual report. So as of 2022, there were 2 soccer fields worth of coral on Bonaire. And then, yeah, if anyone has any questions, feel free to interrupt me. Yes?
So around here, like at Alligator Point and stuff in those bays, or in those like waters, there's these things called artificial reefs, I think, where they like purposely sink something. Because I've gone scuba diving on a lot of them. Like there's barges and there's like pyramids and stuff. How does that factor into this? Do you like combine them, or how does that work?
Yeah. So when people talk about artificial, so reef is kind of like a broad term to mean like structure on the seafloor. So when we say coral reefs, that's like structure made by coral. Artificial reefs is kind of a fancy word for like a lot of times, as you've seen, like cinder blocks. Like it's just like a big thing, and those are mainly for attracting fish. It can be really good at giving fish like a place to, like some habitat, a place to live. Eventually stuff will start to grow in those artificial reefs. So yeah, artificial reefs are like things that are not made out of a living organism. They're made out of something artificial. And they're mainly to draw in fish. Fish love structure and little hidey holes. For coral restoration, people have tried so many different techniques to see like what works best to get the coral to stay out there. So I have that picture of oh wow. So they'll have the nursery trees where they're dangling. Eventually they can, when they're trying to get the coral out on the seafloor, a lot of times when it's sitting in the sand, it might like roll around, or like there might be pathogens or like diseases or bacteria in the sand that will then hurt the coral. So they try and have the coral not directly on the sand. So they will make metal frames and attach the coral to the metal frames. Then they were like, hey, now we're just putting a bunch of metal or plastic into the ocean. What if we made the frames out of bamboo? So they tried making like bamboo frames. They also tried making like hemp like ropes to like have the corals grow on. So there's a bunch of different methods of like things people will put the corals on when they're restoring them. It kind of depends on the environment of like how much water movement there is. Bonaire's doesn't have a ton of water movement usually. They'll have like a slight current but not super strong waves. And so what Reef Renewal has actually found is they can just put these branching staghorn corals directly onto the sand, and within a few months they'll kind of grow and fuse together and just make a staghorn little thicket or bush right in the sand. Some of them do roll away, but the time they save of not having to put all these materials and like build all these structures to put the corals on is a lot, and then they can put that time to growing more corals. So it's kind of like a trade-off. People are trying a lot of different ways to get these corals out there. I'm looking at staghorn. Okay, so this photo here is Reef Renewal's like pride and joy restoration site. It's like 10 years old. This coral like up here is like 6 feet deep. Like it's huge. But this is what the staghorn coral used to look like throughout the Caribbean in the wild. This wasn't just like one singular special site. But it doesn't really look like that anymore. A lot of it looks like this. It drastically declined in the 80s, mostly due to disease, but there are things like storms, the loss of an important sea urchin, pollution, that like ocean warming and bleaching that like really they just never were able to bounce back. And so a lot of it is totally dead rubble like here, or there's like a few live bits here, but they're definitely not 6 feet tall. Like they're not at nearly what they used to be. So they're classified now as critically endangered. And so for my PhD, I really wanted to do a project that combined both advancing like scientifically what we know about corals while also supporting these restoration efforts. So I reached out to Francesca Virdis, who is the director of Reef Renewal Bonaire, and was like, what are some knowledge gaps you see? Like, what would you support you the most and like to scientifically understand to advance your work? And she was like, I want to know how my staghorn coral genotypes are performing after outplanting. So to formally put this into like a more scientific research question, I am asking, is there genotypic variation in fitness traits of outplanted staghorn coral? And so we can study this using a common garden experiment design. And these are a really important experimental design tool where it originated from terrestrial studies where they would have a garden and they would put multiple genotypes of the same plant species into that garden. And because it's a common garden, there's no environmental variation, so they're all experiencing the same amount of sunlight, rainfall, like soil type. And this is important because if there's no environmental variation, but we still see differences among the genotypes, that means it's likely because of genetic differences. And if there's genetic differences, that likely means there's like a gene coding for those traits that the parent could pass down to the offspring. We call this heritability. And heritability is important because this tells us that the corals or the plants or whatever might be able to adapt. So if we find that there is genetic differences in proneness to bleaching or to getting disease, then if that's related to some kind of gene, then we could have a population that is more adapted to this disease that's out there or more adapted to warm waters. So there have been a lot of common garden experiments done with staghorn coral, but they're all at pretty small scale. So like in aquarium tanks or on these nursery trees. And while these are super useful and informative, they don't tell us exactly, they're not exactly representative of what's actually going on the sea floor, where the corals are experiencing many more stressors than what is happening in like an isolated aquarium tank. There's seasonality in certain stressors, like the water gets hotter in the summer. You also have disease outbreaks peaking in the summer. So this is definitely useful in figuring out like a start of what's going on with genotypic variation in the corals, but we're ready to take that next step into looking what actually happens on the seafloor. And so that's what we're trying to do with one of Reef Renewal's restoration sites. So what we're looking at right now is called a photomosaic, and you can think of it as like a quilt of a bunch of photos stitched together looking at this restoration site. It's 50 by 100 feet, so it's like the size of a basketball court about, and it's 20 feet deep, so you can imagine like you're snorkeling in beautiful Bonaire, looking down, and this is kind of like the whole site you might theoretically be able to see, and each of these blue dots is one of the outplanted corals for this study. So just some information on the study. These corals were outplanted in November 2021, so it's a couple years old now. We have 10 different genotypes we're looking at, and there are 9 outplanted corals per genotype, or like 9 replicates. So there's 90 total corals in this study, or like 90 blue dots basically. So to zoom in on what each of those blue dots look like, this is one right here. You have one here, here. It's like all these clusters of the staghorn coral. And this one here is probably like this big. So that's way bigger than what they have in the nursery trees or in the aquariums. So this is super cool. And then one little anecdote I just want to say, sometimes for restoration or research collaboration with restoration, we have like slightly different goals. So my goal for this study is I want to keep track of each of these blue dots, each of these outplanted corals, to know how they're performing over time, which the best way to do that is give them all a little plastic tag that's nice and neatly labeled that I can return to each time and be like, okay, this is genotype number 2, replicate number 1. Those tags get overgrown by stuff really quickly. There's a lot of life on the coral reef, and they also just break off. And it's also putting plastic into the ocean. So Reef Renewal kind of has no interest in that. They don't want to be putting plastic into their restoration sites. They also don't want to be putting in the work to be maintaining these little dinky tags. And so our compromise for this is I basically use this photomosaic as a huge like map. I laminate it and bring it with me underwater. And so I swim up and say, okay, here's this big rock right here. I know I'm right below it. I must be at genotype number 5. And so basically when I go out and survey these sites, most of the time I end up spending is just trying to like read the map and figuring out like where the heck I am in this giant restoration site. Yeah. Once I figure out where I am, I look at a variety of traits related to fitness for the corals. So yeah, just a reminder, fitness is the ability to survive and reproduce. These corals are very slow growing and very slow to reproduce. They're not going to be reproducing in the duration of my PhD, so every trait I'm looking at will be related to survival. So I'm definitely looking at whether each coral just totally survives or dies. But even within a live coral, you can have something called partial tissue mortality. So all of these like orange branches here are live, but these white parts here are dead skeleton, where the skeleton's still there, but it has no live tissue. And so we might say this coral has like 10% partial mortality. I'm also looking at growth, so how much the entire coral changes in size from initial outplanting, like time 0 to 1 year later. And then I'm also looking for evidence of some common stressors on the corals. So like this white spot here is a disease lesion. This is a fireworm that is a big predator of the coral that basically, all this is the worm and this is the coral branch, and it's just like coming through and like swallowing the coral tip whole. We also have ciliate infections, which is this kind of like bread mold looking stuff. These are fish bites. These are from my beloved but complicated farmer damselfish. The idea is that they bite at the coral, the coral gets stressed out, and then algae can overgrow it, and then the damselfish can have expand their algae farm. Yes.
I had a question. When you're down there, how much of your time is focused on just that, and how much is it on, like, hey, there's other cool fish or neat things that you see, or this new worm, or I imagine it's a combination, but I mean, can you really stay that focused while you're down, or is it still really cool to you?
It kind of depends on your approach to field work. Our field work, so I live here like most the entire year, and then we go down to Bonaire for like 2 weeks, and we're diving like every day, like as much as we can. My advisor likes to really squeeze in as much work as we can get in those dives, and you're limited by how much air is in your tank. So our dives might be like 90 minutes to 2 hours. We're usually cranking out as much as we can do. And the way it usually goes is like the first day we'll usually do a bit more of like a get oriented, look at everything, kind of explore dive. And then on the last day we'll do kind of a similar like, you need pretty photos for your presentation, so go take a bunch of pretty photos and pretend you're like with a little clipboard and pretend you're doing science. So it's kind of a combination. Yeah, I mean, so one of my favorite things is that a lot of these staghorn patches that we go to, there's like a group of squid that just sit and hang out. And it's like if you go up to them, they'll run away. But if we're just sitting there working, they get super curious and just kind of sit there floating and watching us. So we have lots of photos of squid. Yes.
Is it like uncomfortable, like diving like that, like so like deep? Like does it like put like going out every day? Does it like put like I guess it would create like pressure? Like is it uncomfortable?
It's exhausting. It's pretty tiring just because like scuba tanks are heavy, and like you put dive weights in and stuff like that. So we're actually at 20 feet, not that deep. People will dive down past like 100 feet. So like you can go down to 20 feet in a swimming pool probably. What I would say, okay, I'm just gonna go on a quick side thing. With diving, you can dive with your tank just has like regular air that we're breathing right now. They also have something called nitrox, which has a higher percent of oxygen in it than what's in air. There's certain depths or durations of dives you might aim for where nitrox makes more sense. Bonaire is a super popular tourist like diving destination, so they have this nitrox everywhere and it's the same price. When you're diving on nitrox every day, you feel way less tired because you're getting a lot more oxygen. So it's just like how if someone's like, I don't know, sick in the hospital, not doing well, they give them extra oxygen. When we dive on nitrox, it's a lot more comfortable, I would say. But yeah, you feel the pressure as you go underwater. You feel the pressure of the water above you, and you like, clear your ears and everything. 20 feet is, we have pretty cushy sights, honestly. Like the water's warm, there's like no current. Yeah, so it can be uncomfortable, but for the most part, this is like the most comfortable the diving can get. Yeah. Forget where I left off. Oh yeah, and then the last stressor I'm looking for is overgrowth. And so the coral can get overgrown by a bunch of different organisms. This red fluffy stuff we're looking at here is cyanobacteria. So it looks like a plant, but it's actually bacteria, like a huge bacteria clump. Okay, so just a couple of results so far. This project is still a work in progress, but I just want to kind of show you like what I've been seeing. So first off, just to summarize what I'm seeing with these stressors, disease is by far the most prevalent stressor. That black bar is the highest at months 3, 6, and 9. And this is important and also makes sense because disease is what caused these corals to decline in the first place. So that disease is still out there. These are kind of considered like a weedy coral species. So their life strategy is more like grow really quickly and then maybe have like a weaker immune system. So this kind of makes sense. The stressors declined over time. They were all pretty much the highest at month 3, which is interesting. So this might say that like the initial outplanting transition from the nursery tree to the sand might be stressful for them, but then once they get settled, they're doing better. And then lastly, there's no damselfish activity. I point this out partly because I really like damselfish, but also it's really interesting. So there's no yellow bars for any of these months. And that's interesting because that's not the case in other places. Like in the Florida Keys, there's stories of like they put these staghorn outplants out, and then the damselfish like come running and flocking to it. They love making their little farms on the staghorn coral. Versus in Bonaire, at this restoration site, there's damselfish like 50 feet away on a wild patch of staghorn coral that have no interest in coming over on the outplants. So it's really interesting. I don't fully know why this is happening, but it's really cool that it's not the same everywhere. There's something different going on here. All right, and the next graphs I'm going to show you now are going to have each of my 10 genotypes, A through J, along the x-axis, and then some trait like growth on the y-axis, and then the trait average will be a gray dotted line. So on the one hand, I might see no genotypic variation. So each genotype is pretty much at that average. On the other hand, you could have the same trait average, but a lot more genotypic variation. So try and, like, for the next graphs, think of which one it more looks like. So this first one is growth. Most of those genotypes are not very close to the average, so we have pretty significant genotypic variation here. I put growth in quotes because all my growth values are actually negative, which means the corals were bigger at the first time point of outplanting versus 1 year later when they measured them. So anyone want to take a stab at guessing as to why my corals seemingly got smaller? Even if you think it's dumb, I want to hear any ideas.
Come on,
Maybe just like pollution and like rising temperatures in the water.
Yeah, it's a good guess. What else we got?
Do corals like shed maybe? Like, you know how like some plants will.
Yeah. I don't think they do. I mean, I could imagine like if one branch was diseased, it might just kind of fall off, and maybe you can call that shedding. Any other ideas?
This might be stupid, but do they like when they have to like build their roots almost or like get like situated in the ground, does that like make them smaller?
Oh, that's a good idea. I don't think so. I think the the biology of coral settlement. So they're sitting in the sand when we put them out there. Even though they look like plants, they don't have roots like they're animals. So they might be, usually they like will sit on something and then they'll kind of grow their skeleton around to attach to whatever hard like rock that they're near. Or sometimes they'll be able to just like anchor themselves more in the sand, although it's not exactly the same thing as roots. These are all great guesses. Do you guys remember Hurricane Ian from a few years ago? Yeah, that's what did it. So I said that Bonaire usually doesn't get hit by hurricanes, but sometimes it still does. It's not 100%. So in between the first time point when we put out the corals and then 1 year later when they measured them, Hurricane Ian came through, which basically means that the corals didn't actually shrink, but because of that strong wave energy, the branches broke off and flew away. But the strength of like it being a common garden experiment is we can assume that each of the corals all experience that same wave force, which means these very big differences in sizes should be due to differences in growth, which is super cool that we can still see that even after a hurricane. Alternatively, I also looked at disease prevalence, so how many of the 9 outplants all had disease or not. There's some variation, like you can see genotypes E and F are pretty far from the average, but a lot of the other genotypes are pretty close to the average. So like we see a bit of variation in disease prevalence, but overall this tells me that there's still disease around there. It's still affecting these corals, and it's at about like a 40% prevalence. So just to circle back to my initial research question, is there a genotypic variation in fitness traits of outplanted staghorn coral? Yes, so far I'm seeing it with growth or like change in size, and then also disease prevalence. So to just come back to this initial list of coral stressors that I showed you, my project mainly comes in and is looking at how disease is affecting these corals and how that relates to genotypic variation. But that's just one piece of the puzzle. There's this whole slew of other things that are affecting corals. So if this is something that you're interested in, there's definitely many things to be studied and figured out about these corals. So with that, yeah, thank you for listening.
Hi everybody, I'm Laura. I'm also a PhD candidate at FSU in the bio department, but I work mainly down at the Coastal and Marine Lab down in St. Teresa. And I work on deep sea corals, so I'll tell you why these corals are a bit different than from what you're probably used to that you can scuba dive and snorkel on like Allie does. But just first, a bit about me. I'm actually from Montana, so I was very landlocked for the first 18 years of my life. But I was that weird kid who decided when I was like 9 years old that I was going to be a marine biologist, even though I had zero idea what that meant. I saw the ocean probably like 3 times before I went to college. But I did get to go to the Oregon coast. My dad really encouraged this dream, and so like he took us on a couple trips to the ocean, and he and I actually got scuba dive certified when I was about 14, because in my brain I was like, you have to scuba dive to be a marine biologist like Allie does. You don't. I haven't gotten diving in years, unfortunately, which is sad. I do love to dive, but I don't have to for my job. But the other cool thing that my dad did is he encouraged me to read books about the ocean. And so this book here was just like a random textbook about the ocean that he found, but it had one single page describing deep sea coral habitats and basically how little we know about them. And so I was like, this is the coolest thing ever when I was 12 years old. So it was always there in the back of my mind, and now it's pretty cool that I'm here talking to you all and telling you about deep sea corals however many years later. But I went to undergrad in Western Washington University. It's a bit north of Seattle, and it was the best place I could have gone. I got really great experiences. And I got an internship actually where I got to go to DC and work with the National Oceanic and Atmospheric Administration for a summer, working with their deep sea coral program. And so that pretty much hooked me. I was like, this is definitely what I want to do. It was very cool. And then I went back to my university after my internship and was really lucky and got to work with a professor who was working on deep sea methane seeps. And so actually out in our Gulf, we have a ton of oil that bubbles up, and animals actually use that the same way plants use sunlight to make food. And so I was really lucky I got to go on a couple deep sea cruises before I graduated undergrad. But I wanted to go back to the corals, so that's why I came here. I work with Dr. Sandra Brook down at the Marine Lab, who traditionally works on deep sea corals. And these corals are a bit different than the ones that you can scuba dive, snorkel on. Like Allie mentioned, one of the big problems they're facing in shallow waters is this coral bleaching, where they actually have this symbiotic algae inside of them that photosynthesizes and gives them a lot of their food. My deep sea corals, though, are past this 200-meter point, where there's no more light in the deep sea, and so they don't need those symbiotic algae anymore. They rely entirely on food that comes from the surface and falls down to them. And you can imagine there's corals actually way down near the bottom of the Marianas Trench. So you can imagine how long it takes food from the surface to get down there and how little actually gets down to the bottom, whereas some of these corals are. However, they form these big, beautiful, amazing habitats that most people don't even realize exist. There's 2 basically big habitat types. One is kind of more of a coral forest that looks like this, that's more of the fan-shaped corals, versus coral reefs that are, actually I have some examples here that I'll pass around. So the coral reef type is mostly what I work on. And so these corals form big clumps and make huge mounds in some places. And this is more calcium carbonate skeleton. It's like a rock, like our skeleton. And some of the more coral forest type corals actually have different types of skeleton. Some have half calcium carbonate like this, but a lot of them have more keratin skeletons. And so I'll pass this around. This is more like your fingernail, actually. It's one of my favorite types of skeletons. But these habitats are really important. They actually hold a lot of commercial species that people fish for that maybe you've eaten, like orange roughy, alfonsino, Oreo fish, golden crab, stuff like that. And they're very biodiverse, actually as biodiverse as shallow water coral reefs. And so these reefs are actually worldwide, but most people don't know they exist. I bet this is the first time a lot of you are hearing that these corals exist, but they're everywhere and really abundant actually around our East Coast and Gulf of Mexico. And they actually made news recently. You might have seen, maybe some of you, that right off of our coast, actually from about Cape Hatteras down to the middle of Florida, it's the world's largest deep sea coral reef. We knew it existed, so the scientists discover is kind of misleading, but we finally measured it, and I think it's 200 kilometers wide, something like that. It's massive. But these global deep sea reefs are very important, but we don't know much about them. Yeah.
So to the shallowest part of that deep sea reef, can you scuba dive?
Oh no, not that one. Yeah. No, these are about 2,000 meters. Yeah, that reef is about 2,000 meters down, so 6,000 feet, right? Am I doing that math right? Yeah.
Do you like send cameras down to like look at?
That's what I'm going to tell you about. So, how on earth do we study the deep sea? So I am really lucky. I've gotten to go on 4 deep sea cruises throughout the time since I've started studying the deep sea. Basically, we take these big ships out into the middle of the ocean, and we can do one of 2 things. We can go in a sub, and I've been really lucky, and I've actually gotten to go on one sub dive in the Gulf of Mexico while I was studying methane seeps. But I did get to see deep sea corals on that dive, and I was pretty excited about it. That dive was about 1,100 meters, so 3,300 feet into the Gulf. The other tool that we can use is basically a big robot camera system, very fancy camera system actually, like 4K video. And both of these options, we can take video of the deep sea, and they have robot arms and like this basket right here we can put samples into and bring them back up. So does anybody in here like to play video games? Yeah. So these robots, these remotely operated vehicles that we send down to the bottom of the ocean actually feed video back up to the ship. And controlling it is actually like a video game. We have a pilot, but sometimes the pilot lets you try it out like I got to do here. And it's like a video game. Like there's a joystick and you have to maintain like the altitude over the seabed, and you have to figure out which direction you're going, and it's very fun. I'm very bad at it. I'm bad at video games. But it's a pretty cool job. And
are you cross training for it now? You got a new
no.
PlayStation or whatever?
But they do actually have like internships for high schoolers to train how to do these things. So it's pretty cool. And then you get to look at the deep sea all day long and go to the middle of the ocean. It's a pretty cool job in my opinion. But what I specifically study about these corals is where the heck the energy goes. They don't get very much food to begin with. This is all they rely on, is just how much they get from the surface. And so where does that energy go? And I want to know mostly what happens with the reproduction, because as with any animal, you kind of put your energy into 2 main processes, either growth and maintenance, or you reproduce. And reproduction actually takes a lot of energy overall. And so overall, there's kind of two big ways that corals reproduce. Basically, you can have a male and a female colony that sends their eggs and sperm up into the water. They meet, they make a baby larvae, it's called, as a baby coral. And then eventually that baby coral settles and becomes an adult, and they can asexually reproduce, which is how we get these big reefs. But there's also this other option that corals can send up sperm, and then a female will catch it and brood the larvae. So they'll keep the baby inside of them like a human does, and then send it up when it's a little bit bigger, or it'll crawl away. And so basically we don't know a lot about corals and how they reproduce in the deep sea because it's so hard to get there. And I actually have a fun example of how hard the pressure is, like how much those subs and those robots have to withstand. So a fun tradition is to put a styrofoam cup outside of those vehicles. And this is an example of what that styrofoam cup looks like after 3,000 meters, so about 9,000 feet. This is the pressure and what happens when you go down there. So it's hard to get down there. It's expensive. We have to take these giant ships and these giant vehicles. So we know really very little about how these corals reproduce. Yeah?
Do you know, like, what the most common form is? I think
the most common form of, like, how they reproduce? Yeah. So we're trying to figure that out. In shallow waters, they're mostly, the reef formers at least, I'll make that clear, they're mostly hermaphroditic, where they have eggs and sperm in one colony, and then, to my understanding, most of them broadcast spawn, so they'll send up the eggs and sperm together. And this is actually really well synchronized, which I'll talk about in a bit. But in shallow waters, we can time it down to like the hour sometimes in some species because they synchronize with lunar cycles. As far as deep sea corals go, I myself actually in my PhD have discovered that, and I'll talk about this, that two species in one of the areas I study are hermaphroditic, and they're not hermaphroditic anywhere else in the world. So we don't know yet what the major way of reproducing is. Yeah, that's a great question, and it's very species dependent. So it depends on like what type of coral we're looking at. It's a great question, but we're still very much learning. And so the way that we learn is the process that I do, because I can't scuba dive down there and stare at the corals until they reproduce. I have to use a process called histology. It's the study of tissues. And I cut up the corals really small into slices that I can put under the microscope. And you can see down here, this is the polyp. This is where the eggs and the sperm are, and this is what it looks like under the scope. These are all big eggs here, and then each of these bright purple dots is a head of sperm. And so I can see what stage they're at, like how close they are to spawning, how big the eggs are the eggs really healthy, stuff like that. And so instead of trying to maintain them in an aquarium, this is actually really hard. Some of our collaborators, like around Norway, are trying to do this. No one has yet successfully used a deep sea coral and spawned it, fertilized it, and then made it settle and grow into a new coral. That hasn't been done at all yet. They have fertilized them, but that's it. So instead, I have to use this process where I take corals from the environment and look at them under the microscope. So I kind of have two major questions that I'm looking at. The first one is they have this really limited energy supply. And so what if we change the amount of food that they're getting or the timing that they're getting that food? What happens to the reproduction? And so this question I'm answering with reefs all across the Atlantic. And these reefs actually spawn at different times, and they get very different amounts of food. And so I'm basically trying to fill in these size classes. And so this is just the size of the egg versus the months. And I'm trying to figure out what environmental variable correlates to when they spawn. Yeah.
So when a fertilized egg is kind of floating around in the water, how far can it travel, before it settles?
That's a great question. It's actually very species dependent. And so I don't know how long they can go, like, survive before they're fertilized. But once they're fertilized and they're that little larvae baby, some of them can go for miles and miles and miles. They can actually, some of them can feed on stuff from the water, the same stuff that the coral would eat normally. But the more they can do that, the more they can eat, the farther they can go. So that's a great question. Yeah. And we think that's why the deep sea is so well connected. And there's species all around the world that are the same. But yeah, I'm basically trying to figure out with this question what correlates to the spawning. And so we think that it's the timing that they get their food, because I can see a difference. There's about like 6 to 7 months in each of these regions before they spawn. So that's one major question I'm looking at. The next one is kind of my pet question, but I look at corals in the North Pacific. And so basically, I won't go in detail, but the North Pacific is actually pretty acidic just because of the way that the currents work and photosynthesis versus respiration, balancing that. It builds up a lot of carbon dioxide in the North Pacific, and that carbon dioxide reacts and makes that water acidic. However, the skeletons should dissolve in acidic waters, the reef formers. That one is calcium carbonate, but just within 10 years ago, they discovered these massive reefs all along this North Pacific region. And so what I'm trying to do is, oh yeah, one last thing. Other big problem is that the deep ocean is also becoming more and more acidic because more and more carbon dioxide is going into the ocean, and this acidity dissolves the skeleton even more. So we want to figure out how healthy these reefs are and what's going to happen to the other reefs that are going to experience this ocean acidification. So basically, I want to know what happens to reproduction when the ocean becomes more acidic and they have to put more energy into building that skeleton because it's actively dissolving as they're trying to build it. And so I can kind of compare the reproduction above the point where it's not too acidic for them and below the point where it is too acidic for them. And basically, I found, like I was saying, something cool where species that are separate sexes everywhere else, I've actually found 3 species that are hermaphroditic in this region and only this region. And so they have, so you can actually see the sperm packet here and eggs. And again, eggs versus sperm. And so I compared this above and below that point where it's too acidic, right? And I can actually see that below that point, or above that point, there's a lot of females and hermaphrodites, not as many strictly males, just making sperm. But below that point, where it's harder to make the skeleton, where they have less energy for reproduction, I actually see about 50% are entirely males. And so this becomes obvious because you can see just to make one egg, it takes a lot of fat, a lot of energy, versus sperm, you can make thousands of sperm for about the same energy input. So there's some possibility here that is worrying that maybe they're turning entirely into male below that point where it's too acidic for them, where they have to work harder to make that skeleton. Yeah.
So I was wondering, I know you're talking about the researchers in Norway who are trying to grow them. How do they mimic like the deep sea pressure in like an aquarium environment?
Such a good question. So these corals actually don't care that much about pressure, but some corals do, and some, especially like deep sea fish, do. So if you want to do experiments with that, there's actually these crazy setups where they have, like, it's like putting it in a vacuum and it presses it. It's really weird. I don't know exactly the physics of how it works, but there are aquaria that they can do that. But these corals actually, especially the ones that they're messing with in Norway, those corals mostly care about temperature. And so this research is especially happening in Norway because they come up really shallow in the fjords of Norway where it's really cold. And that's all they care about. You just have to keep it cold. Yeah, good question. But it's still hard. They're still pretty picky, especially about the temperature. So if they get too hot, like when they're coming up, they're not going to live very long.
Nice.
Yeah. Any other questions? I know I have like 1 minute left.
What's the point of the corals being like different colors if it's like so dark you can't even like see the color?
That is one of my favorite questions. We have no idea. Nothing can see it, presumably. We think it's like the symbiotic bacteria. They don't have algae in them to give them the color, right? But we think it's bacteria. I'm trying to figure out, though, the color is actually passed on to the egg in most of these species. And so I'm trying to figure out if the color affects reproduction at all, because presumably whatever is giving it the color is passed on to the next generation. And so does that affect reproduction? So yeah, we don't know, but they're beautiful habitats. It's just like, why are they colorful? Nothing can see it, we assume. Yeah. Perfect timing. Yeah.